Hello everyone. Today we are going to discuss about an important topic, that is chromatography, which is an important analytical tool in the chemistry laboratory. While the chemists are synthesizing new compounds in the laboratory, this is an important analytical tool which they will use to identify the new compounds that they have synthesized in the lab. So let us go for what is the definition of the chromatography. Chromatography is a way to separate parts of a mixture by using different solubilities of the parts for a mobile phase compared to the stationary phase. Means the components that are present in a mixture can be separated into individual components by the technique called chromatography. So in this chromatography, the two terms that we have to be remember is, that is, mobile phase and the stationary phase. So the mobile phase may be gas or liquid, okay, and the stationary phase may be the apparatus that does not move with the sample. Okay, this is about the chromatography. And if we next move on to the principle, what is the principle that is involved in the chromatography? Okay, in chromatography the compounds are physically separated by distributing themselves between two phases, that is stationary phase and the mobile phase, which flows continuously across the stationary phase. See here, this is the sample that we have injected onto the stationary phase. This packing material acts as a stationary phase, and this is a sample. Means here you can see a number of components, maybe pink, blue and white color. These are the components that are present in the mixture. Now we want to separate these components present in the mixture into individually. So onto the sample we allow the mobile phase. So the mobile phase is a liquid. When the liquid passes through the sample, so the components which are present in the sample, some will be dissolved in the mobile phase and some will adhere tightly to the stationary phase. So the components which adhere tightly to the stationary phase will elute lately, or the components which are readily dissolved in the mobile phase, they elute first. Like that, depending upon the partition coefficient or distribution coefficient of the components between the stationary phase and the mobile phase, these components present in the mixture are separated into the individual components. Means the basic principle which is involved in the chromatography is the distribution coefficient or the partition coefficient. If we next move on to the classification of chromatography, there is classification of chromatography based on different factors. So if you move to the first classification, it is based upon the interaction of the solute to the stationary phase, how the solute molecules are reacting with that of the stationary phase. If the solute molecule are absorbed on the chromatography on the stationary phase, that type of chromatography is called absorption chromatography. Means just the molecules will attach onto the surface of the stationary phase. If if the components that we are taking in the solid phase, if they undergo exchange with that of the stationary phase molecule, then that chromatography is called the ion exchange chromatography. Okay, and what is the next classification? If the molecules present in the solute, if they undergo partition in the stationary phase, that chromatography technique is called partition chromatography. Then the next is the size exclusion chromatography. In size exclusion chromatography, the components that are present in the solute will be separated depending upon the size. Okay, so these are the four techniques. These are the four techniques which comes under the basis of interaction of the solute to that of stationary phase. One is the absorption chromatography and ion exchange chromatography, partition chromatography and size exclusion chromatography. Next type of classification is based upon the chromatographic bed shape. How the bed shape is there. So here there are two types, two dimensional and three dimensional. Again, two dimensional is divided into two types. Those are thin layer chromatography and paper chromatography. So in thin layer, a uniformly coated thin layer of silica gel is used on the TLC plates. Means in thin layer chromatography we will use TLC plates and the stationary phase. It comes under the two dimensional chromatographic technique. And paper chromatography, paper is used as a stationary phase. This also comes under the two-dimensional chromatography technique. And if you go for the three-dimensional column chromatography, okay, and column chromatography, in column chromatography you will use the stationary phase, powdered stationary phase like silica gel, which is a three-dimensional. So column chromatography comes under the three-dimensional. Okay, so depending base on the basis of chromatography bed, chromatography methods are classified into two types. Those are two-dimensional and three-dimensional. In two-dimensional, thin layer chromatography and paper chromatography. In three-dimensional, column chromatography is there. So if you move on to the next type of classification, this classification is based on the physical state of the mobile phase. Either the mobile phase is solid or liquid, a gas. Depending upon the physical state of the mobile phase, the chromatography technique was classified into three types. Those are liquid chromatography, supercritical fluid chromatography, gas chromatography. Means in liquid chromatography, the mobile phase which we are using will be in the form of liquid. The mobile phase will be in the form of liquid. And in the supercritical fluid chromatography, the liquid which we are using is the supercritical fluid. Okay, that is nothing but the supercritical fluid. So if you go for the gas chromatography, the mobile phase which we are using is a gas, either neon or argon, like gases. Gases are used as a mobile phase. This is the brief classification of chromatographic methods. There are three types of classification. One is the classification based on the interaction of the solid molecules with that of stationary phase, and the second is on the basis of chromatographic bed shape, and the third is on the basis of physical state of the mobile phase. Okay, and next, thank you.